Okay, so we can proceed with the session, but let me just brief you about the previous lecture because it needs, reminder because as we are proceeding with the last lecture. So that was our lecture number 8, right? The last Wednesday. And the title of that particular lecture was, I think, Future, right? Future Direction and Emerging Technologies. We already understood and in the personalized medicine actually, and at in that particular session we discussed about artificial intelligence and machine learning in the genomics, also some multi-omics approach we already understood, the meaning of transcriptome and transcriptomics, also proteomics, study of the proteins, and some metabolomics, right? Advances in precision oncology was also the topic of the last session, and some nanotechnology and targeted drug delivery we understood, right? So those are the topic for that was included in your last lecture. And in today's lecture, that is the actually very important in a practical way, as I earlier explained you. And that's what, basically what you are implementing in your daily life, in, implementing genomics into the daily life. Now, this particular session has a very interesting title. It is implementation. Okay, so the title itself has the word implementation of pharmacogenomics in your healthcare system. So we'll see what is the integrating genetic data into further electronic health records, right? You know now the meaning of electronic health records, which is an emerging technology in the healthcare setting, and what actually this genomic setting, is doing here. So basically, you are integrating the patient's genetic data into the health records. Ultimately, when the next clinicians or a physician or any of the prescriber is supposed to give a particular substance or drug maybe to the patient, the electronic health record has some of the information which is genetic information of the patient, which gives alerts, right? So if the patient is maybe to the receiving of warfarin or some of the agent like codeine, right? So this the alert will be there for the physician or the clinicians to use or not to use in a particular patient based on their genetic makeup. So that is our first point, integrating our genetic data into electronic health records. Okay, and the second learning objectives will be pharmacogenomic education for healthcare professionals, because sometime what happens, in my experience also I have seen that many of the clinicians are not aware of the gene drug interaction. Now that's what the exact problem is, right? Because they are not aware of the genetic testing, the interpretation, the implementation techniques, so we cannot proceed further, right? So that is very important to make the clinician, pharmacist, and nursing people aware of the gene drug interaction. Okay, this is the term, this is the future, and it is coming, right? So that is very important. Third learning objective will be cost effectiveness and health economics. Since very first day we have that question in our mind, what is the cost of genetic testing? Okay, many of you might be question as well since the very first second lecture, that what is the cost of genomic testing, genetic testing, and how in low to middle income countries like us, every people cannot afford genetic testing. They are not affording, so some of the population are also there, they are not having a CBC report as well. So the genetic testing is a way beyond thinking, right? Because it costs little bit more. It is costing a 5,000 is a minimum cost, and it goes up to 50,000 whatever test is recommended by the physician. So yes, it is costly. So we'll see the pharmacoeconomical aspect as well today by the real world examples. And last learning objective will be the case studies of the real world application. Okay, so we'll see the United States are practicing, some of the hospitals of United States already practicing integrating this genetic data into electronic health records, and they are receiving alerts as well. We'll see the Indian scenarios as well, what at what step we are heading at, what are the challenges to take care of, because you are the generation who is building further the genomics in future. So basically we can say we should know the challenges. to overcome it, right? We should know the problems where we are going to act, right? So basically the next chapter may be written by you in the genomics. That is what my expectation is, after learning so much of, in detail. So you know the concept now, you know everything. So last lecture is totally dedicated to practical aspect. Okay, so that is a summary of the session. I am sharing the screen with all of you, and as we practiced, you can ask your question at the end of the session. Okay, I hope I am audible enough and the screen also is visible. Just a second, I am sharing the screen. Okay, so I hope the screen is visible. So here we are. This is our course title again, very last time. So basically, that is the pharmacogenomics and personalized medicine. You know now the title. So we are moving forward with the next slide, and it is having lecture number 9 it is. So we already completed 9 sessions and 2.5 months actually. And it is implementation of genomics in healthcare system, pharmacogenomics in healthcare system. These are very clearly learning objectives defined. So very first we have integration of genetic data into electronic health records. Secondly, we'll see the pharmacogenomic education for healthcare professionals. Third, cost effectiveness and health economics. And the fourth topic is case studies of real world applications. I already elaborated the whole 4 learning objectives in detail, right? So we can proceed with the slides. So this is the very first topic to discuss, which is integrating genetic data into electronic health records. Now let me tell you that this session is going to be a 1 hour exact. So it will be and at 7, and this session has only 25 slides, okay, which is quite less as compared to our other sessions. So because it is a miscellaneous topic, but this is very important when you are dealing in a practical scenarios, okay? So this is very important. Please make sure you understand this concept very clearly. Okay, so integrating genetic data into EHR, electronic health records. So you can imagine this. Let's say a doctor is about to prescribe a clopidogrel to a patient post stenting. Okay, stent lagane ke baad, if the doctor is about to prescribe a clopidogrel, what if a pop up, that is nothing but an alert that could arise and tell them or tell the doctor that the patient has CYP to C19 poor metabolizer category status. I really hope that you know now the meaning of this particular sentence, CYP to C19 poor metabolizer, meaning the patient could not digest clopidogrel as expectedly. Okay, meaning the drug won't work as efficiently as it should be. So that's the power of genomics integration into EHR with the decision support. Okay, so you know now whole meaning of this in this particular title, that integrating genetic data into electronic health record. What is our aim behind this is, if the doctor is about to prescribe certain medicines, they will get a pop up, maybe a alert that the specific patient is being sensitized, or maybe some of the category which is not good for the expected drug or ideal treatment. That is what the personalized medicine means, right? So basically, what it is, the definition here is, it is referring to storing patients' genetic test results in nothing but electronic health records. As its name suggests, I hope you know the meaning of electronic health record. The name suggests, and the definition of EHR is exactly in the title. So electronic health records which are saved electronically. Okay, so in a very structured form, very accessible form, and very interpretable format. But I hope now you have the whole concept clear in your mind. We already discussed challenges in particular this aspect, right? So for that we required a very good database to store a large amount of data. Also there is a privacy issue, there is ethical issues, and some legal issues as well. Okay, so it is not as easy as it seems. So basically there is nothing but storing patients genetic results data into EHR, electronic health records, in a very accessible, structured and interpretable format. The goal here is to support real time prescribing decisions, as the yellow line you can see. Okay, it is a real time prescribing decision through what? CDS, Clinical Decision Support Tool. Okay, so that is the exact aim behind integrating genetic data into electronic health records. I hope it is making sense. So, how exactly it works? So maybe genetic variants like CYP2C19, TPMT, HLA-B 5701, it is a variant, right? So see, you know now the meaning of this, but I am repeating again. CYP2C19 family of CYP450 enzymes, 90% of your drugs are going to be metabolized by CYP family, right? TPMT is a phase 2 enzyme, and HLA-B also should be checked if you are prescribing certain medicines, right? Because of the hypersensitivity it may lead. Antiviral agents are associated with hypersensitivity if this HLA-B 5701 variant is being found, right? We know ample of examples associated with this three. So maybe the genetic variants like this are being stored as discrete data, and when a doctor prescribes a drug affected by those genes, the system gives CDS, which is a clinical decisions support system, right? Gives alert. Now, what it can actually do, it can recommended dose change because a patient is categorized as poor metabolizer or maybe the rapid one. We need to adjust dose. Now we know the concept, right? So CDS can actually recommend the dose changes, also suggest alternative drugs, and also give you the warning of some serious ADR risk if the patient had any link between this. So this is the aim behind integrating our genetic data with our electronic health records. It is not just only putting the data into electronic system. We need this. Okay, we need the recommendation of dose change, we need alternative drug options, and we also need warning signs of ADR and risk. So this makes a therapy very very comforting, right? Very very personalized maybe. So this is what the actual meaning of this. Why it matters? So basically it can actually prevent adverse drug reaction. So obviously if the patient is about to receive clopidogrel after just having a stent, so and if the patient is having poor metabolizer category, so the clopidogrel won't work, right? And in fact adverse drug reaction of clopidogrel is very very large, on a large scale it is. So it can actually prevent ADR, it can optimize your overall efficacy, and it brings actionable pharmacogenomics into the routine care. So that is what our aim is behind integrating your genetic data into electronic health records. If someone asks you about what is the future, what are you aiming of कि कैसा system होना चाहिए in the prescribing, so you can always say like this, the patient's genetic data should stored in the electronic health records, and some CDS alert should be there. This is not impossible because it is practice in the United States. Many hospitals are doing it. Obviously they are having some of the very strict act and very strict laws to follow this genetic data security. They are having some of the best laws. I think in many of the legal ethical issue session we discussed about the HIPAA, right? Many of GINA, those are the act which are make sure this genetic variability. So that is must before proceeding with this particular conditions, right? So what are the challenges is the very first challenge we have. The particular challenge we have, that is data interoperability. Now this is very important. Data interoperability. So labs use some various formats like PDFs, some unstructured notes to give the genetic interpretation. Electronic health records further struggle to interpret or act on this data. Also, we need some standards like HL7FHIR genomics to fix this. Now, what is this? This HL7 FEAR is the pronunciation. HLR FEAR, that is FHIR. The full form is Fast Healthcare Interoperability Resources. I am again repeating. FHIR stands for Fast Healthcare Interoperability Resources. You know the meaning because as its name suggests, it is nothing but giving you the resource, or the you can say it is giving you the platform to share the data interoperability के साथ, right? It is a flexible, providing a resource which is very flexible to change or to interchange the genetic data. So we need standards like this which is basically it is for exchanging healthcare information electronically, HL7. And it is next generation framework which is developed by Health Level 7 International, that is HL7, to further facilitate interoperability between different healthcare system. If I can say the meaning of interoperability, so this FHIR aims further to solve the challenges of getting different healthcare system to communicate and share data effectively. That is what the meaning is. So ultimately we want to exchange the data from lab to particular hospital, or hospital to hospital again. So we need some standards like this which is being practiced in other developed countries. So data interoperability is the first challenge which is restricting us to integrate genetic data into electronic health records. Second challenge we have, that is a security and privacy. As I earlier said, DNA data is highly sensitive. It is far more personal than only, as compared to cholesterol or some blood sugar level data. So genetic is very, very sensitive data. It requires stronger encryption to control access and patient consent mechanism, which is very hard to take. Okay, because they might be having some other issues, some legal ethical issues will be there. So security and privacy is another challenge to integrate our data into electronic health records. And the third challenge we have, that is a dynamic updates. Now this pharmacogenomic knowledge evolves, okay, and a gene drug interaction considered harmless today may become significant tomorrow. So that is what tomorrow bringing, right? Dynamic updates. Systems must reanalyze stored genetic data when the new evidences are emerges, and it is emerging in every single day. So if you are keeping electronic health record of your genetic data, of your patient's genetic data, you need to continuously reanalyze it, okay, and update it, because that is very important. Otherwise, it may gives you a false results. So these three are the challenges, which is restrictions of our electronic health records integration. Okay, so these are some of the examples we are having here. So we have real world example here. The St. Jude Children's Research Hospital in United States, that includes a program in their hospital which is PG4, that is pharmacogenomics actually, and kids, KDS. Okay, so pharmacogenetics for kids, the program itself has name. And genetic data from over 14 key pharmacogenes is primitively collected for pediatric cancer patients. So this is the study that was being performed, and the program was being launched in the United States hospital. Result integrated into the hospital's EHR. Pediatric cancer patients has the sample patients, and they have collected 14 key genes, right? And then that result integrated further into the hospital's own electronic health records. Further, a CDS, clinical decision system, alert further warns the clinicians when the drug like 6-mercaptopurine, the immune suppressants, or some kind of TPMT variant, if the present in the child, they are making alerts with this. Okay, so any kind of pediatric population receiving 6-mercaptopurine is ordered for a child with TPMT variant. So this further helps to adjust the dose or choose some alternative to prevent severe neutropenia, which is associated with 6-mercaptopurine if the patient is having TPMT variant. And trust me, this if the severe neutropenia occurs in the child, it may lead to directly child death. Okay, because this is very serious matter. So this particular hospital is associated with those kind of integrated system. Okay, so what is the impact of this particular example? It is improving patient safety, personalized dosing, and model now adopted by other hospitals as well. So that was the first data was performed in the United States, but you can see how they are evolving, right? So that was a real world example. Now we are moving forward with the future tools and innovations which are coming. So, what is coming? Smart on FHIR genomics. Now, what is this smart? So, this is nothing but a set of open standards and some specifications that enable your healthcare applications to securely access and exchange the data from electronic health records using your FHIR, that is a fast healthcare interoperability resource standards. So ultimately, SMART is nothing but it is an open standard, okay, and a specifications it is, and that enables your healthcare application to securely access and exchange the data between ESHR and FHIR, right? So it is a modern open standard for integrating your genomic apps with electronic health records. It is enables tools that can plug in the genetic data and further guide the prescribing from within any of the electronic health record system. So this is upcoming, right? You can see the title. It is a future tool. Many loops are there in smart, but we can have a look that how exactly the system is working. It is lab to lab interchange, or is it hospital to lab interchange? It is giving you the accessible and the special specifications to exchange your genetic data among the different resources, okay. Next we have the future tool here is the genomic passports, okay. The genomic passport is a new term, very important it is. You can have idea that the patient is having a the card or e-card basically, that includes all of its genetic details into it, okay. So that they can carry anywhere, if they are traveling, if they are moving from country to country, they are having that with them, the genomic the details. So basically genomic passport is a concept, often it is associated with other things as well, that refers to a standardized way to represent and manage the access rights and permission for genomic data, okay. It essentially act as a digital identity, as I earlier said, and also it can act as an authorization token, okay, that allow the researchers to access specific data sets based on the predefined criteria. So that is a genomic passport. That is also the new upcoming technology and the innovation. Also we have cloud APIs. Now API is application programming interfaces. API, application programming interfaces, okay. So this cloud API in the context of genomic refers to API that are hosted on a cloud platform to further facilitate access and interaction with the genomic data and the tools. And this API further enables researchers to perform tasks like the data retrieval, analysis, and the workflow execution in a distributed and a scalable manner, I can say. Right? So that is the importance of and the aim behind Cloud API. But let me tell you, you can imagine if the patient is having genomic passport or a cloud API, still we have many challenges to overcome in this particular future innovations. But this must be, if you are aware of, many companies are also working on the emed card, right? So basically a person is having just like the other card, if the person is having also the medication card. So that is also the upcoming innovation and tools, but in context of genomics we are having some smart technology, genomic passport, and the cloud APIs which are upcoming, okay. So this is what we are heading at. So that is the first topic, first learning objective is clear, which is integrating your genetic data into electronic health records. Moving forward with the second learning objectives for the last session is pharmacogenomics education for healthcare professionals. As I earlier explained, many clinicians are itself not aware of gene drug interaction. See here, the greatest barrier to implementing genomics isn't the technology, trust me. It's whether the clinicians know what to do with the results, right? So this is very important. So it requires a skillful person, right? So if the genetic test result shows that CYP2C19 poor metabolizer, but the person itself is not aware of the interpretation of result, what to do? That is what the meaning of education, okay. So despite growing access to the genetic testing, many healthcare professionals further misinterpret the pharmacogenomics reports, okay. So misinterpretation leads to the false prescription, lack of confidence in prescribing based on genetic data because of lack knowledge, worry about ethical and legal issues as well. You know the ethical dilemmas. We already discussed, a whole lecture was dedicated on ethical, legal, and some social issues. So some people are also worried about these ethical and legal issues as well. Okay, so this is the challenge. So why it's so critical? Because lack of training is resulting further in the hesitation in adaptation. Pharmacogenomics is not yet fully integrated into most of the medical or pharmacy school curriculum. That is very important, right? Genomic is a new term. Okay, so it is not integrated fully. Many clinicians haven't even heard of the key, that is a gene drug interaction, as I earlier explained you, right? Second thing to understand here is a complex reports need interpretation skills. So PGX results are not positive or negative like the infections test. Infection test results can be identified by any of the individuals, right? Maybe of non-medical background as well, because the normal range are written already in the report, so you can interpret it at at some extent. But when it comes to the genetic test result, you require a skill. Okay, as I earlier explained you, reports say things like CYP2D6 intermediate metabolizer, which require what? Contextual decision. What do you do later, right, if the patient is having intermediate category of metabolizing, right? So that is also the challenge. And the third is ethical dilemmas and patient communication. Like, it is hard to explaining genetic risk to patient. It is hard to handling incidental findings and ensuring privacy and confidentiality on a second hand. So this is why the education to healthcare team is very important on the context of genomics. So who are the key stakeholders here and what are their roles? So we have doctors, pharmacists, genetic counselors, nurses, right? And the role in PGX, the pharmacogenomics, if we can focus on doctors, the role is selecting, testing, making prescribing decisions, okay? And the role of pharmacists, please, all eyes on the role of pharmacist. You can see here interpreting pharmacogenomics results. So if you are a pharmacist or becoming a pharmacist, please know this is your responsibility to interpret pharmacogenomics results. That is the exact reason behind this course. Okay, so you can see here the role of pharmacist here is interpretation of results, dose adjustments, and substitutions as well. Okay, so here we are also having the category of genetic counseling, but the genetic counselor, the role itself is very less in the India right now, but it is in a growing stage. Okay, so the role of there is to communicating risk, next consent, and the family implications. And the role of nurses here is monitoring the outcome, ensuring that patients understanding. So that are the teamwork, right? So basically, pharmacogenomics is a team sport, and each player needs training in their own role. So the reason behind putting this course to make yourself aware of the genomic term. Okay, and I'm very glad that you have understood thoroughly the concept of pharmacogenomics. Very important it is, right? Let's move forward with the real world example of UF Health. So, see here, the University of Florida, UF Health personalized medicine program, they have been launched, and it is implemented a pharmacogenomic education series alongside some electronic health record integration. That's what the lack in India. So they offered some of the CME. CME meaning continuing medical education. Okay, CME, continuing medical education, which is accredited modules on how to interpret the genetic results, when to test, and how to act on findings. That is very important, right? So first of all, you need to understand to interpret the result. Next is when to test and when to not, and how to act on finding. If you are finding anything, what is the next step is very important. So the result of this particular launching the program is over 90% of physicians are reported confidence in applying pharmacogenomics post training, and adoption of pharmacogenomics alerts are also there in the electronic health records, and that is improved significantly. You can think that if we are just imagine if you are solving every aspect of integration, and you are further integrating genetic data in electronic health record, everything is smooth. Electronic health record are giving you the signal signs alerts. But what if your staff is not accurate enough in not having knowledge enough to what to do next, how to act on the findings, what is the scenario? So that is simultaneously should be the CME should be there, the program should be there, the education should be there. So this is a prime example of importance of education plus infrastructure, and then you can go for the real clinical use of pharmacogenomics. So it is very complex, and we are going into very detail that, at a very very root level, what is restricting us. So that is the strategies to improve the education. What you can do? Curriculum integration. So include more pharmacogenomic concepts in MBBS, maybe in PharmD, MSc, nursing curriculum. Case-based teaching should be there for better context you are learning. That's what we are doing, right? Next, you can also go for some online CME module, continuing medical education. For example, CPIC training module. I think I have included the link also, so that afterwards, after completing this particular course, if you have still interest and eager to, the more interest is there, you can go on the site and check the CPIC training module. Okay, I hope now you know CPIC, what it is. It is a guideline, right? And some of the NIH, that is National Institute of Health resources, for example G2C2, that gives you some of the genetics and genomics competency centers. So they are also having some continuous medication education modules, so that may be used. Maybe you can go for simulated PGX reports. So use mock reports, what we are having here in the practical sessions, to train the clinicians in interpretation for the genotyping and phenotyping drug relationships. And last but not the least, institutional grand rounds or workshops should be there. Okay, so monthly PGX sessions for clinicians or maybe some other healthcare professionals should be there, featuring some real cases, discussion on new CPIC guidelines, and some local implementation strategies as well, how to overcome. So this may be helping you further to strategize your, educational strategies. So this is very important. Moving forward with our third learning objective, which is cost effectiveness and health economics in pharmacogenomics. So this is very important topic again. I have a question. Would you spend 10,000 rupees on a genetic test today itself to avoid? This is the one question, that would you spend 10,000 on a genetic test? The maybe 90 answers from 100 is out of 100 is no. But understand the second question. To avoid a 1 lakh rupee hospital admission due to a drug reaction next month. The answer will be yes. Okay, so please understand the question. Would you spend 10,000 on a genetic test today to avoid 1 lakh hospital admissions due to drug reactions next month? Very important question. I think some of you might be having, okay, so you are replying. So this is what the meaning of cost effectiveness, right? So this is the heart of the economic debate around the pharmacogenomics. It is always there. So here is the very first thing to understand, the economic debate, right? So yes, genetic tests cost money upfront very largely, as since the very beginning of the session I elaborate, right? Often it may have $60 to $300 or more maybe, which is in Indian rupee it is a 5,000 to 25,000, and it goes up to 50,000 as well. Okay, but what it can do on other sides, it can prevent adverse drug reaction which cost maybe more than the the money which is written above. Also, it can reduce failed therapies or rehospitalization. Also, it can shorten the time to effective treatment. This is very understandable and clear, right? So, the real question is, actually, the real question is not is the test expensive, but the real question is what is the cost of not doing the test? So this is exactly practice you can also adopt to convince your patients in case of treatment failure, because we are not going with the every prescription genetic testing, but in case of multiple treatment failure, in case of resistance, in case of other things, you can obviously go for this aspect of genetic testing. Okay, so the real question is what is the cost of not doing the test? So let's understand this also, which is a key scenario again and key health economic concept it is. We have two things to understand here. The very first is a QALY. QALY stands for Quality Adjusted Life Year. Now, sometime, it is not as important, but I have included the slide because sometime in some of the statistics and, articles which is being published online, in which you will be finding this word QALY. So that is the only reason I have put into the slides to understand the genomic concept. So there is a quality adjusted life year, okay, which is nothing but a measure combining life expectancy and a quality of life. So for example, a year in a perfect health is a 1 QALY, one quality adjusted life year. But a 1 year with some of the disability is reduced, 0.6 QALY, quality adjusted life year. So obviously, if you are having a 1 QALY year full of perfect health, you are good on a scale, and if it is less, you are on a bad scale, right? So that is the ultimate meaning of QALY. Now see the second one, which is a ICER. ICER is an incremental cost effectiveness ratio. Now, this particular term compares the cost of a new interventions, basically a genetic testing cost, to the usual care. Okay, so ICER is equal to cost of PGX minus cost of standard care, which is divided by QALY's gain with PGX, जिन्होंने PGX मतलब फार्माकोजेनोमिक्स इन्वॉल्व किया है, एंड QALY's gain score minus QALY's with the standard care. Now if you are integrating this particular formula, you will be having a particular, the digit, and a low ICER is a good value for money, basically. So if your math is good, you will be solving this particular puzzle, but do not go into that detail. But ultimately understand that this quality adjusted life year and incremental cost effective ratio is showing that what should be done, okay, and what should be ideally done, whether you go for test or not. So generally an intervention is considered cost effective if it costs less than $50,000 per QLY, quality adjusted life year. So this is overall in the United States, or adjusted per country's GDP. This particular QLY is being adjusted as per the country's GDP. So in India, we can say if you are spending 20,000 rupees per year to your overall health, so that is also considered as a cost effective. Okay, so if cost is less than 20,000, that is being seen as a quality adjusted life year. So that is another health economic concept to learn when it comes to the pharmacoeconomics. Okay, so I hope it is clear. And the third we have, that is a case-based example to learn this. So for example, abacavir for HIV. Now this is an antiviral agent which is being prescribed widely to cure as a, the HIV. So we need to check HLA-B5701 genotyping variant. Okay, further helps to avoid severe hypersensitivity. Now see, the test cost of this particular HLA-B is $60. Maybe it may vary like between $60 to $150. And ADR, if you are prescribing abacavir without HLA-B test for HIV, so the chances of ADR hospitalization, which is can exceed up to $5,000 to $10,000. Imagine the difference. So the verdict here is highly cost effective it is. What is cost effective? To test HLA-B 5701 genotyping variant before prescribing abacavir into HIV patient. That is being highly cost effective. Okay, this is now routine in the United States, Europe. Exactly what it's making a difference between developing countries and developed country, right? So also we have second example to understand here, that is a warfarin dosing. So for warfarin, what you need to check? You need to check CYP2C9, the enzyme which is responsible to metabolize your warfarin. Also you need to check VQRC1, right? Vitamin K epoxide reductase, too, because it is also important in case of warfarin metabolism. So this particular thing further promise safer and personalized dosing. But because in case of decreased poor metabolizing category, what it needs, warfarin accumulation. Okay, so internal bleeding will be there. If it is toxicity, like it is a fast metabolizer, you won't get the effect. So maybe the exact for exact cause you are prescribing which is not achieved. So that is what the warfarin is having, right? Narrow therapeutic index. So but the clinical trials are there, for example COG, didn't consistently show better outcomes than the clinical dose adjustment nowadays. So it is now used in the selected cases only. So what, if you can observe, like in every ICU patient has that warfarin in it, right? So we have that ward rounds. We collected so many of cases, and we know that not every patient is going for CYP2C9 and VQR7 test. Not even 1 in 10, I think. Okay, so this is particularly used in very selected cases, in very complex anticoagulation, liver damage, and very elderly patient is if it's the case. So this is a case-based example to understand the genetic testing. And the last we have, that is a TPMT testing before thiopurines. That is also the example we already understood in the previous sessions. So basically, this is used in the leukemia or any kind of autoimmune condition where thiopurines are being used. TPMT is a phase 2 enzyme which is responsible for metabolizing your drugs like thiopurine, 6-mercaptopurine also. And TPMT variant is present, so it is very high risk for myelosuppression, right? So that is the exact mechanism for pediatric cancer hospital, which is in the United States, is working on, right? So also it is genotyping or enzyme assay, which can avoid toxicities if you are going for TPMT testing before thiopurine. Cost effectivities, especially in the pediatric oncology, and now it is a standard practice in many pediatric cancer centers. So this is being widely used. Okay, so my point behind putting this example is you need to understand what is commonly being practiced now and what is into the future. Okay, so you can work on the problem nerve. Okay, so that is what the TPMT. Now we are moving forward with the global perspective. So what is in the high income countries? For example, in United States, in UK, Netherlands, and Singapore, they are already integrated the pharmacogenomics into national insurance programs, which is a Medicare, because that is also the issue. I think you remember the ethical social issues. Then we also have that issue in insurance, right? So maybe some of my insurance not covered your genetic test expense. Okay, so that needs to be carried out, and this particularly the high income countries are already integrated genomics data into national insurance program under Medicare. If you are aware of this program, very good. If you're not, it is widely being practiced in high income countries. So electronic health record plus clinical decision supports alerts, so it is giving you the ultimate therapy, right? So primitive testing programs like 120K genomes is being used in the UK. So these programs are more focused on the integration of genetic data into your currently existed system. Okay, and on another hand, we have low and middle income countries. So the challenge obviously is a cost. Okay, so cost of widespread PGX testing is very, very large. Solution is only one. Use a targeted PGX testing, targeted. Okay, so only in the high risk drugs. That's what we can do nowadays. Carbapazim, or some warfarin, thiopurine, right? These are the areas where the treatment failures are happening. So at that thing, you can go for genetic testing or pharmacogenomic testing, right? Or for the patients with major adverse drug reaction and the treatment failures, as I earlier explained. So in India, targeted PGX testing that could prevent deadliest ADR with minimal upfront investment, especially in oncology in an HIV care. So what is the scenario in India? I hope now it is clear. Moving forward with another thing. So, the ultimate summary of this is pharmacogenomics is not just a scientific innovation. Please understand, it is an economic opportunity as well. It offers the healthcare system the chance to move from reactive care to predictive care. So, one size fits all approach abhi ja raha hai. It is vanishing, and some of the more personalized predictive care is coming. But like any investment, it requires smart planning, careful evaluation, and context specific strategies to implement this. This one line has lots of meaning. Okay. So that is what the meaning is. Moving forward with the fourth number of learning objective, the last one. And these are having some of the case studies of real world application where exactly these things are happening. So it's one thing to understand the science of PGX, right? But how is it really being used in hospital? That is we need to understand, like how exactly it is being used. So let's explore the two powerful real world stories from the US and right here in India. We have two scenarios to understand. First, we'll see the United States study so that we can compare it with our own country. So, case one here is a Mayo Clinic's right protocol, United States. Let's understand what they did. They launched the right protocol, right. So the right drug, right dose, right time, rational use of the drug, right? And they enrolled 5,000 patients for primitive genotyping, which is focused on clinically relevant genes like CYP26, TPMT, SLCO1B1. I hope now you know SLCO1B transporter based, right? So it is actually responsible to transport from your gut to the liver, right? So if you are having a variation in this, maybe it is leading to again many of the categories, poor metabolizer, maybe, right? So focus on clinically relevant genes like this. Results are integrated further into the electronic health records, and clinical decision supports alerts are built in for the gene drug interactions. So that is what they have practiced. Outcome kya aaya? So outcome, you can see over 20% of patients are triggered actionable PGX alerts when the new medications were being prescribed. For example, patient about to receive a codeine, CDS flagged CYP2D6 poor metabolizer. So what happens next? Doctor avoids ineffective therapy. So instead of codeine, they will prescribe some alternate agents. Okay, that doesn't require CYP2D6. So instead of ordering genetic test during a crisis, which is not possible all the time, this Mayo build PGX into routine care before the drug is ever prescribed. So you can imagine this slide is very important, right? You can also be the first one to integrate these things into your practical learning, or your practical whatever you have been practiced. So this is what they are doing. ऐसे ही नहीं कहा जाता कि they are way ahead of in the healthcare, but these are the systems they are integrating. Okay, what is restricting us, I don't know, but some of the restrictions are there, like we need some strong act if you are going with the genomic testing and everything, right? So we know the challenges. So let's see the Indian scenario where the Med Genome and Strand Life Sciences are working together. So if you don't know, this Med Genome and Strand Life Sciences are both prominent genomic and bioinformatic companies, which is based in India. But they have distinct focus and the areas of expertise. First, MedGenome is a diagnostic and research company specializing in genetic testing for various diseases, including some of the rare inherited diseases like cancer, reproductive health पे काम करते हैं लोग. And the second one, that is a Strand Life Sciences, on the other hand, is a bioinformatics company that develops software and provides service for genomic data analysis and precision medicine with a strong focus on in silico technologies. So two companies are there. They have different perspective, like the area of expertise, but the aim is same. Okay, so what they are doing, what is happening in India, MedGenome and Strands Life Sciences offer further the pharmacogenomic test panels to some of the cardiologists for clopidogrel and CYP2C19 to check, some psychiatrists for some selectively serotonin reuptake inhibitors and CYP2D6 and 19 again, some of the oncologists for tyrosine kinase inhibitors and EGFR, ALK and TPMT studies. So they are providing panels to understand these concepts, how they can implement this concept in India. Tests are available at a major hospital chains like Apollo, Fortis, Aster and Manipal also. So this is very important to understand what is happening in India. What is the model? So the test ordered by the clinicians or by the hospital pathology labs. Some hospital integrate pharmacogenomics into the tumor, the boards or the personalized therapy plans, and growing awareness in the urban metro centers, especially for oncology and cardiology. So this is where the exact India stands. Okay, we are working, but we are still under pipeline to make this a real. What are the challenges in India? Still out of pocket in most of the cases, like not covered by the insurance is the major issue. Limited use in the rural and low resources settings. Lack of national guidelines. We don't, as I earlier said, we need to have some specific guidelines. Okay, if some of the issues may arise, you need to have guidelines with you. But academic interest is growing. So that is where exactly it is. What is the takeaway? So India is moving fast, but in a fragmented and market driven way. With more institutional support, with more pharmacogenomics can scale to reach to the millions. So that is challenge and the takeaway from the message. So that is the end of the session, guys. And what are the points to take home for today is integrating genetic data into electronic health records, pharmacogenomic education for healthcare professional, cost effectiveness and health economics, and the last was case studies for the real world application from United States and India. So that was the overall session. So I hope it is fruitful, and I'm waiting for your feedback also. Okay, and I think someone has message. Yes, ma'am, but education is most important. Awareness about genomic in population because cost of test more than yes, exactly. It is precisely rightly said. So that was the issue and challenges and the whole genomic. Right. Give me a 1 to 2 minutes, and we'll be back to the question and answer scenarios. Okay. Okay. So any of you may be having a question. Okay, good evening. Can have this happen just like vaccination, protect against future disease. Genomic testing done early in life or after condition like stroke can guide lifelong personalized treatment. Exactly, that's what the meaning of integrating genetic data into electronic health records, and that is the exact aim behind genomic passport and cloud API we discussed in this session, right? So that is exactly the aim that it can be or it can done earliest if possible and integrated further into the own person's own something which should be there, right? It helps choose the right drugs and avoid. Yes, exactly. So any of you might be having question, please put into the chat box. Also, you can unmute yourself if you want. I think there is no question, but I want to brief you all on something because it is the last session. So, so here we are, right? After 9 amazing sessions and nearly about 2.5 months, right? So it was 9 amazing sessions and 2.5 months of deep diving into the world of pharmacogenomics and personalized medicine. Okay, so I would be grateful if you are giving a feedback and if you are attending your MCQ test. First of all, a big round of applause to you all for staying committed, curious, and open minded throughout the sessions. We started this journey talking about genes, drug responses, variability, and how medicine is no longer one size fits all. And by now, I hope you all realize just how powerful this field is, not just on the paper, not just for exams, but in real clinical practice, right? So, from tailoring antidepressants based on CYP450 genotyping to predicting chemotherapy as well, the toxicity, and to understanding why that one patient never ever responded to the standard drug, because that is exactly happening daily life, on a daily life in whenever we are collecting cases or wherever we are on a ward round, right? So that is the exact reason behind why that one patient never respond the standard treatment. Maybe the answer lies in our genes, right? So this is the future, and guess what, the future is already here. It is just needs you to carry it forward. So I know lecture ends, but the assignments also wrap up, but learning really never stops. So the real power lies in applying what you have learned in your internships, in your hospital rounds, while talking to the patient, while researching, or even while simply observing patterns in the prescriptions. So here is my little request to all of you, that don't let this knowledge sit idle. Use it, practice it, talk about it, teach it, and apply it. And that's how it becomes a part of who you are as a healthcare professionals. Okay. Also, this isn't a goodbye. We have got more exciting topics and online courses coming up for you. Basically, we have planned a lot of things for the near future. The goal is nothing but bridging the gap between the textbook learning and the real world impact. Okay. So if this course spark something in you, stay connected. Keep learning, keep growing. Before I sign off, just remember one thing, that every prescription you handle, every patient you meet, it's an opportunity to personalize, to optimize, and make a real difference in the treatment. That's the power of pharmacogenomics, and that's your powers too. Okay. Thank you for being part of this journey. I'm very grateful and genuinely hope to see you all in the next sessions. Okay. Upcoming courses as well. So again, if you have any doubt, you can connect at any time. You can simply put into the WhatsApp chat. Okay. So just remember that Climate is only a one message away. Okay, someone has one question last, I think. Ma'am, genomic can help in psychiatric disorders. Please give an example, because at present time, psychiatric treatment run on the basis of hypothesis. Exactly, they are assuming the treatments, right? So basically, I have that example of SSRIs, so selective serotonin reuptake inhibitors. If you are using it, so it is having a major gene drug interaction. The chances of interaction with CYP2D, to C19. So basically, that needs to be addressed, but still we are under pipeline in many of the settings. You know the Indian scenario now. The two companies are working on it. They are suggesting the psychiatrist as well to go forward at the genetic test, but the scenario is very different. Like many of clinicians, many of the healthcare professionals are still not aware of the gene drug interaction, right? So this is very important. Actually, we as a healthcare professional didn't see any of the in our curriculum as well. The no term is there like a pharmacogenomics or a genetic testing. It should be there. Thank you for your feedback. We know the importance of genomics and personalized medicine. It is possible to make it affordable in future so that it can available easily for all. It is possible, but on a I hope that we as a next generation can focus on this. And I'm very impressed by this question, because exactly that what is restricting us is a cost. Okay, so we can have the alternatives, but as I earlier said, it is a novel thing, and it is emerging in the field of healthcare. So there are some restrictions, but we can solve it. Now the problem, the challenges. You can come up with the solutions as well. Okay. So that is what the meaning is. Thank you guys for joining once again, and all the best for future. Thank you.